Very happy
Okay,
to
you're
have
up,
the
Aaron. Go for it.
opportunity to join you today virtually and present an update to Project Hope, our endeavor to understand the evolution of pediatric glioma under therapy. Project Hope and Project CARE are part of a nationwide consortium of labs funded by the National Cancer Institute and the National Brain Tumor Society to assemble single-cell atlases based upon longitudinal glioma specimens. My lab at UCSF and Mariella Philbin's lab at Dana-Farber have been tasked with single-cell library prep of specimens collected both at our institutions, but also drawn from CHOP and Stanford. We've also done quite a bit of analysis of those data and expanded our cohorts from the original HOPE cohort. And I'd like to share some of that analysis with you today. So to give you a little bit of overview, Project Hope's goals are to profile archival tumor specimens from pediatric high-grade gliomas. And we use single nucleus RNA sequencing for this purpose. My lab was an early adopter of the single cell RNA-seq technology. And in those early days, it was a limitation that we had to work with fresh tissue. But with the advent of approaches to extract nuclei, intact nuclei from frozen tissues, that really allowed us to open up the archives and to assemble cohorts that had greater statistical power. Our focus was on paired longitudinal samples as well as hard to acquire samples. And we sequenced samples with two different modalities, the 10x genomics modality, which gives you a large number of cells, but relatively low transcript coverage, and the SmartSeq 2 plate-based technology, which sequences fewer cells, but you get very deep transcript coverage, allowing you to analyze expressed mutations, alternative splicing, and some of those questions in a deeper way. Now, the overall question we want to answer is, what's the effect of standard of care therapy on tumor composition at recurrence? What about the evolution of the tumor microenvironment? By way of rationale, consider the different mechanisms by which intratumoral heterogeneity can be a barrier to therapy. And there are essentially two. The first is at the genetic level. You might have a pre-existing mutation which is resistant to a given therapy. For example, an EGFR mutation providing resistance to RTK inhibition. And then that particular clone may grow out after treatment to recapitulate a completely resistant tumor. Likewise, there may be some tumor evolution, genetic evolution taking place where de novo mutations occur to provide resistance. The second type of resistance, Howard Fine here refers to it as epigenetic resistance. I would call it phenotypic resistance, cellular phenotypic resistance. There may be different cell types within the tumor, and some of them may be preferentially sensitive or resistant to therapy. We've seen a high degree of plasticity in glioma cells, and so there may be the ability for different cells to convert between different cell types, transitioning to a resistant state to overcome, to handle the selection pressure of therapy, and then perhaps transitioning back once that pressure is relieved. Well, what about genetic resistance? Recently, the GLASS Consortium has assembled a very large cohort to address this question of longitudinal specimens, 288 patients treated at 35 different hospitals. These are two key figures from one of their publications. I won't go into details in the interest of time, but I will just say that they found evidence of selection pressure only in shared mutations, but not in mutations which are private to recurrence, and in IDH wild type GBM. And you can really see that in this first panel, where they show the ratio of non-synonymous to synonymous mutations. One would expect it to be positive if there were any selection pressure. And these results suggest that the clonal architecture observed in the initial disease mostly persisted into recurrence, that the strongest selection forces occur early in gliomagenesis, but therapy itself doesn't provide a strong selection pressure at the level of genetics. In my mind, this would dovetail with the analyses of Christina Curtis at Stanford, who analyzes and models the diversification of mutations within different cancer types. She has a big bang model of evolution where there's an initial diversification of genetics, but then those clones are remarkably stable throughout the course of the disease for many different types of cancer. And this analysis, to my interpretation, would support that for glioblastoma as well. So that gives us a motivation to try and understand the phenotypic heterogeneity, the phenotypic evolution, and how cellular phenotypes and plasticity can contribute to resistance, in addition to genetics, perhaps biased by genetics, but not necessarily dependent upon genetics. This is the core HOPE cohort, 11 pairs of high-grade gliomas. The majority are from young children or adolescents, all frozen tissue that we profiled with single nucleus RNA-seq, as well as select modalities on sub-cohorts. SMART-seq2 and 10X modalities were applied. And we also profiled 10 diffuse midline glioma specimens. I'd now like to tell you a little bit about the analysis we've done of the data that we've generated. And in the Philbin lab, they've spent a lot of time focused on diffuse midline glioma. I'll touch on some of their work at the end, and I'll show you now some of our work on high-grade glioma. Now, we've used the funds that we received for Project HOPE. We've leveraged them, and we were able to grow our cohorts and also analyze other types of specimens. Here at UCSF, we do a large number of surgeries for adult glioblastoma. So we were able to acquire an archival cohort here of adult glioblastomas, as well as adolescent and young adult AYA glioblastomas, which are paired. So primary treatment naive specimens and paired recurrent specimens. For our AYA and adult populations, all of the patients have been treated only with standard of care, temozolomide, radiation, surgical resection. So relatively homogeneous from that perspective. We combined that with our pediatric high-grade glioma specimens, and we now want to understand the differences we see between primary and recurrent in terms of composition, and what of that correlates with age or tumor anatomical location. In this overview analysis, this is an analysis of all the cells. Each dot corresponds to a cell. You've probably seen this type of single cell data representation before, about 500,000 cells in total. We've separated the great thing about single cell RNA-seq is that you can separate the tumor cells, the neoplastic cells, which harbor clonal mutations, from the non-malignant cells using an analysis of expressed mutations. We've done that here. And then the non-malignant cells, over the years, we've developed techniques to classify them in terms of cell type. And in these specimens, we observed quite a few non-malignant oligodendrocytes, but also astrocytes, neurons, T cells, and monocytic lineage cells of different types. So in terms of a comparison of composition, primary tumors in pediatric and AYA populations were actually very similar to adult GBMs, with very few statistically significant differences. Most of our specimens had a relatively high level of tumor purity, and we didn't see any statistically significant differences between the composition of the microenvironment when we compared pediatric, AYA, and adult cases in terms of the primary tumor. At recurrence, however, we do see a lower purity in adult and AYA specimens than in the pediatric cases, as well as, and that goes along with larger numbers of oligodendrocytes and neurons within those AYA and adult cases. Now, one of the things my lab is interested in is cellular hierarchies in different types of brain tumors, especially in high-grade gliomas. And we've done some work to assess the cellular phenotypes of cells in the glioma microenvironment and their relationships. This is a meta-analysis of data from our lab, just the primary adult and AYA cases I just showed you, combined with data from the Neftel et al. And Couturier et al. Papers, recent single-cell studies that also profile glioblastomas. we find that glioblastoma neoplastic cells reside on a single axis of variation, ranging from the Verhoek mesenchymal to Verhoek proneural phenotypes. Now, Dr. Camara gave a very nice explanation of the mesenchymal phenotype and its origins historically, so I won't recapitulate that. I'll just add that underscore that the mesenchymal phenotype is associated with invasiveness, an inflammatory microenvironment, as well as radiation resistance. We did a lot of work to try and understand the mesenchymal phenotype. Despite being studied in the literature for roughly a dozen years, the upstream regulators of the mesenchymal phenotype are still incompletely understood. Whether the mesenchymal phenotype is cell intrinsic or whether it's inducible by factors from the tumor microenvironment is still an open question. In our analysis of adult specimens, we performed single cell RNA-seq, but also single cell ATAC-seq on a relatively large cohort of these specimens. And this single cell ATAC-seq assay, that allows us to identify regions of open chromatin and the transcription factors that bind them. Taken together, we can combine the two data modalities, and this approach identifies cis-regulatory enhancers, as well as the transcription factors that binds them. It also tells us what transcription factors are utilized in a specific condition, such as primary versus recurrent or pediatric versus adult. When we analyze neoplastic cells only, these are only neoplastic cells you see in these figures, we essentially identified two clusters of transcription factors which were being used in the neoplastic cells. The first was comprised of neuro-D and neuro-G proteins, olig-1 and 2, sort of a canonical family of proneural transcription factors that might be typically expressed in a glial progenitor cell, but the presence of neuro-D and neuro-G also implies neurogenic capacity. The second cluster of transcription factors are the AP1 transcription factors, again, dovetailing with the remarks made by Dr. Camara, FOS and Jun proteins. We see an increase in their utilization at recurrence, and we identify the core mesenchymal hallmark genes as being downstream of this AP1 complex. So AP1 transcription factors bind local enhancers from our analysis in a variety of genes, hallmarks of the mesenchymal signature, as well as the master regulator of inflammation and of kappa B, regulator matricellular proteins associated with invasiveness, genes associated with the radiation response and radiation resistance, DNA checkpoint genes, whose elevation can provide resistance to ionizing radiation. In our adult cases, we see a pronounced shift in the frequency of mesenchymal cells between primary and recurrent cases. So we see a marked increase from less than 25% to over 50% of the cells being from mesenchymal cases. This is a paired test. In each individual patient, we see an increase in the percentage of cells. We don't see a statistically significant increase in the percentage of cycling cells. So we conclude that at least some of the increase in the presence of mesenchymal cells is coming from an increase in the birth rate of the mesenchymal population. And that dovetails with an increase in the percentage of mesenchymal cells that we estimate to be in the cell cycle. Intriguingly, when we look at the pediatric and AYA data, although we do see a mesenchymal shift in the AYA population, we don't see a statistically significant shift, or really even any perceived shift in the pediatric population. So I'd like to begin to wrap up here, and I just want to make a quick remark about the works from the Philbin lab. They focused on diffuse midline gliomas with H3K27 mutations. They were able to assemble 50 diffuse midline gliomas, ages 2 to 68 years, so really a remarkable cohort. They profiled with single cell RNA and ATAC-seq. I won't go into the details of their presentation in the interest of time, but their work is soon to be published, and I would point you to that. Thank you so much for your time.